question and answer session will follow the formal presentation. If anyone should require operator assistance during the conference, please press star zero on your telephone keypad. As a reminder, this conference is being recorded. It is now my pleasure to introduce your host. Thank you, Kwang Duo. You may begin.
Good morning, everyone, and thank you for joining us this morning for this conference call. My name is Kwang Do. I'm the President and CEO of BioVie. I am joined today by Dr. Joseph Palumbo, who is our Chief Medical Officer, and Dr. Stephen Arnold of Harvard Medical School and Mass General Hospital. Dr. Arnold was not involved in the trial that we will be discussing today, but Dr. Arnold is a member of our Scientific Advisory Board and has provided valuable advice and critique of what we do to help us get better. Right. So we are here today to discuss materials that we presented at CTAD last week. Right. And as you know, the clinical trial in Alzheimer's disease is one of the leading conferences in this area. The data we presented there has been well received. And we wanted to have this conference call to further explain and clarify how we see the data and why we're cautiously optimistic about this trial. Okay. So I will not read for you this safe harbor statement, but just to make sure that everybody understands that we are making certain forward-looking statements today during today's call. First, a few words about what we presented. As I mentioned, CTAD is one of the leading scientific conferences in the Alzheimer's research world. We made our first podium presentation at CTAD last year, and we were glad to have been given another one of these very few podium presentation slots again this year. And since we are fundamentally a science-led organization, we always want to take the opportunity to engage with other leading scientists about this work, our work, right? And CTAD is perhaps the leading place to be able to do so. Furthermore, we are the company that is furthest along in clinical trials in Alzheimer's disease, working on inflammation and metabolic pathways. And therefore, we believe the data that we have is valuable to contribute to the understanding of the disease and what our drug candidate can do. Right? At CTAD, we presented blinded data that pulled together placebo and patients who were treated with NE3107. This is a standard approach taken when companies discuss data before they are unblinded. Right? And we had hoped to have had unblinded data at CTAD, to be honest, but CTAD moved a month earlier this year compared to last year, and thus we could not get the data completed, cleaned, verified, and unblinded in time for CTAD. So we presented what we had. So we presented the data that we had as of October 18th from roughly 322 subjects whose data were verified or in the process of being verified and cleaned as of this date. Our team is still in the process of verifying the data, and we're working very hard to get to database lock and unblinding later on this month. I should always note that as we unblind the data, as we clean the data, what we found in this presentation may always change. So as we move towards the data, I would like to deliver the key message here, which is you have to consider the totality of the data and not any one factor to really interpret and understand what's going on. This is how the scientific community does it, and it's certainly how the FDA will do it. It's when we present to the FDA, they will look at the totality of the data, right, and not any one particular measure. And in looking at the totality of the data, we conclude that NE3107 appears to be biologically active, and that it appears to be having an impact on the cognitive, the biomarkers, and the imaging endpoints that we've looked at in the trial. So first, to start off with the cognitive and functional assessments. In the clinic, clinicians use measures such as ADAS-Cog, CDR Sum of Box, MMSE, and so forth as a clinical assessment of the subject's abilities. Right? And as we look at this pattern, we have some optimism that there is an effect, a drug effect going on. We make that comment because if there were not a drug effect, you would expect all the subjects to be clumped together and that they would be roughly around the no change line or it's somewhat worsening, somewhat getting better if there's a learning or a placebo effect. But the fact that we see a pattern of many people getting better and many people getting worse would suggest to us that there is something going on beyond just a group, the drug not working. You would not see a lot of people getting a lot better if there is not something going on here, right? And the magnitude of the quote-unquote getting better from this blinded data is a little too large from what you would expect from just a placebo effect. The other thing that I look at particularly is what's at the bottom of this page, which is the correlations. People who are getting better or worse on one metric tend to be getting better or worse on the other metrics as well. So I interpret this as there is something really fundamentally going on underlying this, right? And it's not just purely random. That right, if it were random, people could be getting better on one metric and not getting worse on another, right? So this gives this pattern, this consistent pattern of improvements and worsening across all these cognitive and functional assessments, give us the first indication that there's something interesting going on with our drug candidate. Another interesting indication has to do with the biomarkers. And we were very excited to see the results on this page. Let me explain why. From a mechanistic standpoint, we understand that NE3107 reduces the TNF-alpha burden, right? So blocks inflammation. And by blocking inflammation, you would then also expect a blocking of the production of the amyloid precursor protein, or APP. And with a lower level of APP, you would expect to see a lower level of buildup of more amyloid plaques. And the belief in the community is if you have lower inflammation and lower additional APP burden, microglia and the natural body's natural defenses will have a chance of essentially removing some of the plaques that have already built up there. And so that's from a mechanistic biochemical perspective of what 3107 could be doing. So over a long period of time, you would expect that by reducing TNF-alpha, you would also have an impact on amyloid burden as well. What we did not expect was to see this impact within 30 weeks. We measured the amyloid burden using a blood test from a company called C2N, called the PrecivityAD test. The PrecivityAD test is the only FDA-approved blood-based test that measures the amyloid burden. The PrecivityAD test has been shown to be well correlated with the amyloid burden if it had been measured by PET. And the part that we found exciting is that if you look at baseline before treatment, there were 63 subjects that we had data for at baseline who were shown to be positive amyloid burden as measured by the PrecivityAD test, and 103 who were considered to be negative. And then when you take the same blood test and measure it again 30 weeks later, over 1/3 of those subjects that were positive at baseline had a reduction of their amyloid burden so that they were considered to be amyloid negative after 30 weeks. Now, this to me is really, really exciting because I have never seen a subject have a reduction of amyloid without some kind of intervention. Right? And so this gives us a second indication that there is a drug effect that potentially is going on, right? And this is heading in the right direction, directionally, of what you would expect if the drug was having an effect. And furthermore, we had a small imaging sub-study that uses FDG PET, right? And FDG PET, the way this is conducted, has also been shown to be correlated to what's going on with amyloid burden. And we saw a large proportion of these subjects in this small sub-study see improvements, i.e. reductions of the amyloid burden. So the fact that we saw a reduction of amyloid burden from this group of people gives us cautious optimism that there is something going on here other than a placebo effect, other than that the drug candidate is not working. We also had another small imaging sub-study looking at volumetric MRI, and here we saw an increase in the volume into hippocampus and the amygdala, which also is very encouraging information as well. Right? And lastly, we look at what's going on with inflammation and the metabolic measures. We know that 3107 focuses on reducing TNF-alpha. And here we see a beautiful pattern of scattering of TNF-alpha. Right? So we see reductions in TNF-alpha, and we see worsening or increases in inflammation. As far as I know, there is a small possibility that untreated subjects could see a reduction of their TNF-alpha if they dramatically somehow change their life around, dramatically change their diets, and so forth. Right? But absent a remarkable, miraculous change in some way, you would not expect to see this kind of changes in the reduction of TNF-alpha. And on the right-hand side, we also saw an increase in beta cell function and insulin signaling. Again, this goes at the hallmark of how this drug is expected to work. So in totality, when I looked at this data, I see an impact on cognitive functions, I see an impact on biomarkers, and I see an impact on imaging, right? And the impact is of such a nature that you would not expect to have seen it if there was not something going on fundamentally with this patient, fundamentally suggesting of a drug effect. So let me just pause here and turn it over to my friend Joe Palumbo just to see if I missed anything that we should have mentioned.
I think that you've done just about everything that's necessary to explain the data. I just wanted to go back, however, and just talk about the trial design a little bit and the sensitivity of the trial design. So I think you've mentioned that when we look at the accuracy, the precision of the study, we're looking for markers moving in parallel when they should move in parallel. But we're also looking for evidence that the study is proceeding with the right patients and that they are able to show progression of disease in a short time scale. So if we go back to slide number 5, please, which is the amyloid burden with the branching diagram. If you look at this baseline versus week 30, I think it's important to understand that we deliberately in this study did not enrich specifically for patients who were A beta 4240 positive. We collected that data, but based on our hypotheses, we think that we will have a broader effect in this larger population than simply focusing on those people who would meet inclusion criteria for other studies of amyloid-related therapies. So, of course, this was a wonderful discovery to see that we do appear to be able to engage that. And we'll know more when we unblind, of course. But what I wanted to point out was the folks who started out as negative. So that's the N equals 103 in the bottom part of that slide. And what you're looking here for is the fact that you do have a shift towards positivity. That means that the patients who started the study with the phenotype, the symptoms that present to the doctor's office, that those people actually have the right diagnosis and that they proceed to show this shift towards amyloid positivity. So it tells us that we've captured the right individuals for the study. It shows us in the course of only 6 months, people who start out as negative shift towards positive. It gives us a good look at the sensitivity of the study. Right? And remember, half of these folks are on drug and half are on placebo. So for me, this is a signal I was looking for. In the midst of COVID, in the midst of everything else, did we capture the right patients and would they progress as expected? Right? And you wouldn't necessarily see this in a study that was only looking at positive patients. at baseline. Now, the data and positivity is great, of course, that we have people who shift towards this negative status, and that is absolutely exciting. But there's also this validation that the study is long enough, at least to me, in looking at this shift from negative to positive in a 6-month basis of the study. And so I'm actually very happy with that. And that was all I really wanted to say, Con.
All right, terrific. Thank you, Joe. Let me now ask Dr. Arnold to comment, given he has been steeped in this for decades and really understands this data, this field. So, Dr. Arnold, any reactions, any thoughts on the data that we've just presented? You may be on mute, Dr. Arnold. We cannot hear you.
I'm sorry. I was on mute. Thank you, Con. Can you hear me now?
Yes, we can.
Yes.
Okay, great. Okay, so it's nice to speak with y'all. I was encouraged by a few aspects of this data. The things that really stand out to me, were some of the positive movement that we see in the the cognitive data, as well as the metabolism and inflammatory markers. The amyloid, I'm not sure what to make of. I mean, I think it's nice to see. We'll have to see how that unfolds. But I think what has impressed me about this molecule all along has been the focus on inflammatory and metabolic mechanisms as opposed to amyloid. Amyloid itself, while we define the disorder or the disease by the presence of amyloid in the brain, it's not highly correlated with disease progression or severity of illness. You can have a head full of amyloid, but still have relatively normal cognition. But what the difference is between the people that actually progress into cognitive impairment and dementia versus those that just have, benign amyloid in their brain, if you will, is really the inflammation. When you look under the microscope at brain tissue in people with Alzheimer's disease who are demented versus Alzheimer's disease, meaning amyloid plaques and tangles in the brain, who have mild symptoms or no symptoms, the difference is that the people with dementia have abundant inflammation and significant metabolic disturbance, insulin resistance, and other aspects of neuronal metabolic dysfunction. So to me, looking at those signals that we're seeing in the FDG PET, and then the cognition, and then the TNF alpha and other inflammatory cytokines, those are sort of like the promising leads. Of course, we're blind to everything. We don't know who's changing, who's not changing at this point, but still, those are the promising leads. And then, I do have to say that ultimately we're interested in neurodegeneration. And the volumetric MRI scan measures showing some hippocampal, actually either stability or enlargement, I think are highly significant as well. So, just in sum, I do think that there are some, if we're trying to read the tea leaves in an unblinded way, or in a blinded way here, I'm seeing reason for encouragement.
All right, thank you, Dr. Arnold. Let me just also go back and just tackle a couple of questions I've been asked that may be on the minds of some people on this call. So let me just start there, and then we'll open up some Q&As. Right. A question I've been received is, if you look at all these functional and cognitive measures, are there ones that are more important than others? Are there ones that we should focus on more than others? Right. And to me, the two most important ones on this page are ADAS-Cog and ADS and CDR Sum of Box for the following reasons. One is that they are the most frequently used metrics in the field. Most researchers would use one or both of these. And then I like them because, frankly, the FDA likes them, right? 2 drugs have been approved on CDR sum of boxes so far, along with the changes in amyloid plaque, right? And so as we approach data unblinding, please recognize that we have the ability to modify our protocol and to modify our statistical analysis plan almost to the very end before we unblind. And of course, that's what we are working very hard to do. We are finalizing discussions with our IRB and the FDA on what our ultimate endpoints will be, right? And we will submit that in due course, and we will use that to guide the unblinding process and ultimately how the trial will be evaluated in terms of its primary endpoints. Right, so that's question number 1 that I've been asked. Question number 2 is what kinds of distributions, as on this chart, for example, should we expect? Right? And the answer to that is that there is no answer to that. We don't really look at distributions per se. What we're looking at are just patterns, the spreading patterns of people getting better or worse, right? Because if there's no drug effect, everybody would clump together. If there's perfect drug effect and perfect worsening, everybody will be on opposite ends. And how quickly people migrate from the middle towards the ends represent, to me, an indication of how quickly they are worsening because they're not on drug, or how quickly they are potentially improving if they are on drug. Right? And I've been doing this thing long enough to know that you can never expect everything to be perfect when you unblind. Perfect would be everybody that improved were on drug and everybody who got worse were in placebo, right? It never works out that way. So unblinding will tell us ultimately, but I'm encouraged by this pattern that I see on this particular page. Right, with that, let me stop here, and operator, if we could open it up for a few questions, please.
Thank you. We will now be conducting a question and answer session. If you would like to ask a question, please press star 1 on your telephone keypad. A confirmation tone will indicate your line is in the question queue. You may press star 2 if you would like to remove your question from the queue. For participants using speaker equipment, it may be necessary to pick up your handset before pressing the star keys. One moment, please, while we pull for questions. Our first question comes from Jay Olson from Oppenheimer & Company. Jay, please proceed.
Oh, hey, congrats on the CTAD presentation, and thank you for presenting this update today. I have a question for Dr. Arnold. Along the lines of what Kwong was describing, could you, Dr. Arnold, please describe what you would expect in terms of a 6-month cognitive decline and also changes in PET imaging, MRI, and other important biomarkers in the placebo group so that we can try to imagine what might be happening in the active NE3107 treated group?
Sure. Well, so, one of the things that I think why you can, so typically, CDR and ADAS-Cog, they change relatively slowly, and the earlier you are in the AD spectrum dementia process, the slower the change. So if this were an MCI mild AD, very mild AD trial, as is most commonly done nowadays, the placebo group, most people would not really change in any kind of measurable way. I think one of the nice things about the design of this trial, for being a short trial, is that you are in the more dynamic range of the disease. So the mild, moderate disease is where you can get, the kind of sweet spot for things like ADAS-Cog to be able to detect change. So, I think you're correct. 6 months is a little bit, it's short. Certainly 12 is better. 18 is even better. But for a 6-month trial, to maximize the sensitivity, I think the patient population that they chose is the right one. And you could see some movement. I think, the way for MRI technology now, 6 months is probably, where you can start to detect differences in hippocampal volume. So I think that's good. And then for some of the other outcome measures, certainly, FDG PET or cytokines or maybe even amyloid levels amyloid levels. These are more dynamic, and you would be able to detect some change. So I think I went a little bit beyond your question for the strictly clinical CDR ADAS-Cog type of things, but those are my thoughts in general about the changes that you might be able to perceive or see in this group in 6 months.
Great, that's super helpful. And maybe if I could ask a follow-on, because there is such a wealth of data here in terms of cognitive measures, imaging, and biomarkers, could you just talk about which part of this blinded data you find most intriguing?
So again, I think that I do like to look at distributions a lot, and to see some of the long tails, and including people improving dramatically, is kind of curious and interesting to me. One of my gripes about the way a lot of trials report out is that, they give an average and a standard deviation, and you're never really sure who's changing or who's what. Showing the data like this, including the long tails and the people that are changing, I think can be very, very informative. And some of the things that we see, I would not expect unless there were some kind of biological effect.
Okay, great, thank you. And then maybe if I could, one last question. As we look ahead to the upcoming unblinded data, can you just describe what you would envision as sort of a best case scenario? And then assuming that we can achieve that best case scenario, what would you recommend as next steps for the study of NE3107?
Yeah, so that's I'm encouraged by all of this, actually, and the clinical, of course, is the most the most important at the end of the day. But in a study of several hundred people for 6 months, whether there is a clear, if there is a clear cognitive signal in CDR, that's really quite profound and profoundly important. But really, I would be pleased if we saw, a change in the metabolic, a good solid change in the metabolic activity in the FDG PET. I would be really encouraged by the inflammatory profile that we see with the patients. So, those are the things that I would be looking towards, because I think that those are the things that will be most movable within 6 months with this drug. And if I were to see positive signals from them, then to me that would be strong rationale to move on. And then in terms of developing it further, certainly larger sample size, more diverse. I think I'm also very interested in this compound for, we like to think of dementia as, one thing, Alzheimer's disease, but really, there's a tremendous amount of heterogeneity within Alzheimer's disease itself, but also some of the other associated related disorders, like, the Parkinson's related disorders. And we're learning, day to day how important some of those other features are. So I would advocate for extending it into some other neurodegenerative, more diverse neurodegenerative disorders.
Great, that's super helpful. Thank you so much for sharing your insights, and we look forward to future updates. Thanks for taking all of our questions.
Sure. Thanks.
Jay, it's Con here. Let me just add upon what Dr. Arnold said for a minute. In terms of the trial itself, as you may recall, this trial was initially powered to have 80% powering with just 125 patients in each arm, right? So that's why initially this trial was only intended to enroll about 320 patients, right? And we just went for a larger sample because we just saw an acceleration of enrollment towards the end. So in terms of what to expect as we go forward, right, let me just bring up this slide here. This trial was our first phase 3 in mild to moderate Alzheimer's. And our default plan is to conduct a second phase 3 on a global basis so that we can file the drug around the world. Right? That second phase 3 in mild to moderate is slated for sometime in the middle of next year. Right? But based upon what we saw in our phase 2 trial, we will endeavor to launch a phase 3 in NCI early part of next year. That trial will also have a short 6-month time frame to primary endpoint, but will have an open label extension out to 12, possibly 18 months so that we can demonstrate disease modification, right? So I think what we're trying to get to is 6 months to get an indication, a primary endpoint around symptomatic relief, and then use the open label extension in a subsequent study to demonstrate disease modification. We're just being very practical, if you will, given the fact that we're a small company with limited resources, and we just want to make the resources go as far as they can. Right? And in terms of what to expect, we are currently working busily to verify, clean the data so that we can lock the database. And if everything goes according to plan, we hope to have top-line data reveal, the unblinded data, after Thanksgiving, early December sometime. Right, so operator, do we have any other calls? Any other questions?
Our next question comes from Robin Karnakas from Truist. Robin, please proceed.
I guess I have two. One for Dr. Arnold. What delta on CDR sum of the boxes at a 6-month time point would you find clinically meaningful if that's what they choose to take forward in the statistical plan? And then how would you incorporate this drug into practice if we're on the market? I know would you add it on top of things or use it as a first-line drug? And then I have a follow-up for Kong.
Yeah, so, I tend to avoid, what is the cut point for what would be significant, significant, because I do like to look at all of the data, as kind of Kong mentioned. But the way I would see it is that, I do think we're headed towards combination therapies, and that this is, adds significant value to a therapeutic regimen if it, holds up in terms of showing its improvement in inflammation, metabolism, and degeneration. So I would see it, if anti-amyloid therapy improves and becomes less burdensome and less costly and, more accessible to people, I could see this as an add-on to that for Alzheimer's disease. And I also would, clearly see it as a single agent disease modifier as well.
Thank you. That's helpful. And Kong, what options are on the table for changing the statistical analysis plan? Can you just go a little bit more into depth as to how you're thinking about that?
Sure. Thanks, Robin, for that terrific question. As you know, we enrolled a very heterogeneous population here, right? So we have some people who are considered to be A beta positive, as well as those who are A beta negative. We have people who are insulin resistant versus those who are not. We have mild versus moderates, and so forth. So our team has taken great pains to predefine a series of subpopulations, right? where we think that we may be able to demonstrate a drug effect versus compared to placebo. Right? So all the different metrics that we are able to gather, but from a biomarker standpoint, we're trying to create different subpopulations around that. So to manage expectations here, right? I do not expect us to have a total a great home run out of this thing, whereby 3107 is going to be effective for all subpopulations that we assess and that we dramatically reverse cognitive decline and so forth. It will be great if we see that. That will be a grand slam home run. That will be a great advancement for the community. But the probability of that happening, I would say, is probably small. I would be very, very happy with one or more subgroups whereby we demonstrate a slowing of cognitive decline equal to or better than what the monoclonals have been able to show, right? But because if we are able to do that, that in and of itself is a win, right? And I think in the clinics, the clinicians and the patients would value this drug because we know that this is a very safe drug, and we know it's a oral compound, so you don't have to worry about transfusions, you don't have to worry about injections, you don't have to worry about ARIA and brain bleedings and so forth. So a non-inferior in terms of efficacy will get us a win because of the superiority on safety. Now, I know I'm very explicitly setting a low floor in terms of expectations, right? I always like to set low expectations and then over-deliver if we can, right? So the way to think about it, a win, one subgroup, non-inferiority to MABs in terms of efficacy, and we'll win on the superiority of the safety, right? And I think hopefully we'll be able to over-deliver on that. Does that answer your question, Robin?
That's really helpful. And then just taking it one step forward, in that scenario, that setting the low bar scenario, like how will the FDA view this data, and what then would be required to get on the market? Would you be able to incorporate this data in some way, or would you think there's a risk of having to run 2 phase 3s?
Well, our default plan is we're going to have to run another phase 3, right? So let me just set expectations there, right?
Would you have to run 2 or just 1?
I think we are optimistic that this will count as one of the 2 phase 3s that we would have to run, right? And so our default plan is that we would have to run another phase 3 in mild to moderate. We will run that trial on a global basis, and that trial would support both the U.S. and the international filings, right? But as we unblind the data, and if we get strong data on both the cognitive measures and we have an impact on A beta, as we are seeing a signal here, that will open up an opportunity for us to go and have a conversation with the FDA about accelerated approval, right? Because that's actually how aducanumab and lecanemab were approved, right? Again, if I were a betting person, do I say would I bet on that being the outcome? No, that would be crazy to do so, right? But I think there's always a possibility that could emerge.
Okay, and then just going back to the two phase 3s, because it seems like, if with Lilly they had a positive phase 2 or somewhat positive phase 2, it didn't fully hit, and then a real positive phase 3. So, so how comfortable are you that the FDA is fine with that if you don't hit in the overall population? Or is that, you think that the FDA is just getting more comfortable with having a phase 2 that's okay in some way, and then a phase 3, and then you can still get full approval? Just where's your confidence level lie with the FDA? I think a lot of our confidence levels are very low at the FDA lately, so just want to get your confidence.
Well, I'm not going to endeavor to speak for the FDA, right? But I think but we are quite hopeful that this study, if it unblinds the way that we expect, that it would be a strong study in front of the FDA when it gets there.
Okay, great. Well, thank you very much for taking my questions.
Thank you, Robin. All right, let me answer one other question that I'm surprised no one has asked, actually. I've heard this question before, and which is, We have shown in today's presentation impact on the cognitive measure, ADAS-Cog, CDR-SB. We have also shown data on changes in inflammation. So the obvious question, of course, is what's the correlation between those metrics, right? And because it's the obvious question, we did not look at that. We did not want to take any steps that would potentially lead us into unintentionally unblinding this data. We believe this is an important trial. We want to maintain the absolute integrity of the trial and live within the rules of how these trials are done so that when this information does go to the FDA, the scientific integrity is maintained, and that's the reason why we did not look specifically at the correlation of the one thing that we know that the drug may be affecting, inflammation, and correlating that directly with our endpoints, right? So we will let the data speak for itself when the unblinding takes place, but we did not want to take any steps along the way that could lead us into unintentionally doing something that we did not want to do, right? So with that, I know that we are over time here. Let me close the call by saying that we are cautiously optimistic when looking at the totality of the data. Right? And I do not believe you can look at any one particular measure to say things are working or not working. You have to look at the totality of the data. And the totality of the data here would suggest that any 3107 appears to have an underlying biological effect, and that biological effect appears to be having an impact on the cognitive, functional, biomarker, and imaging endpoints that we are looking at in this trial. So we are cautiously optimistic that when we unblind the data after Thanksgiving or early December, that this could potentially lead to a significant improvement, a significant new tool for the community to treat Alzheimer's disease when it's fully registered. So with that, I thank you for your time. This presentation is now available from our website, and the replay will be available within the next 24 hours or so as soon as we get it back from our partners. Thank you very much for your time. Have a good day.
This concludes our question and answer session. This also concludes today's teleconference. You may disconnect your lines at this time. Thank you for your participation.